Welcome to our first annual Miss Genetics USA pageant. Today we begin our questionnaire section. First up is Miss Abby Smith. She's going to tell us a little bit about DNA replication.
The process of copying DNA is semi-conserved, which means that the DNA is half conserved during this process. In general, DNA is replicated after the unwinding of the double helix. A replication bubble will form at this point where the replication takes place. DNA polymerase will add deoxyribonucleoside triphosphates to the growing 3' end of the strand. DNA can only replicate from the 5' to 3' direction, and a new strand of DNA will be produced. There's a beta replication, which occurs in prokaryotes. An example would be in E. coli. This occurs because they have circular DNA, and it's a bidirectional process which will produce two circular DNA molecules. There's also rolling circle replication, which occurs in viruses and in the F plasmid of bacteria. This requires the cleavage of DNA, which when one of the circular strands breaks apart. This process will produce is unidirectional and produces one linear molecule that can circularize. There's also eukaryotic replication, which occurs in linear double-stranded DNA. This process is bidirectional and they have multiple origins of replication. Initiated proteins will unwind the DNA, and helicase will break the bonds between the two strands. The single-stranded binding proteins prevent the formation of secondary structures in the DNA, and DNA gyrase will control the supercoiling of the DNA. An RNA primer is also needed for the initiation. Since DNA polymerase can only add from the 3' end, there's both continuous and discontinuous replication. This produces a leading strand and a lagging strand. The lagging strand produces fragments called Okazaki fragments that are later linked together into one long strand by DNA ligase. At termination, the RNA primer is released and a gap is filled by telomerase.
Welcome back to the Miss Genetics USA pageant. Thanks for watching, and tonight Ms. Smith is going to tell us about the process of transcription.
Transcription is the process of copying DNA into a complementary strand of RNA. We need a DNA template, a ribonucleotide triphosphates, and a transcription apparatus. In prokaryotes, a consensus sequence is needed to serve as a binding site for the sigma factor. At initiation, the promoter is recognized, and then in elongation, the polymerase moves downstream, unwinding the DNA and elongating the RNA from 5' to 3'. In termination, the RNA polymerase reaches a termination sequence, which is either row-dependent or row-independent. In eukaryotes, we have to modify the chromatin structure in order for the DNA to be accessible to transcription machinery. It's a similar process, but there's no sigma factor. Instead, there is a core promoter and a regulatory promoter. The transcription factor recognizes a consensus sequence called the Tau box on the DNA template. Then elongation proceeds the same as in prokaryotes. This process produces pre-mRNA, which can go through RNA editing before being translated.
Welcome back for our final questionnaire session. Tonight we'll ask about translation.
Not everything that is transcribed will be translated. Only mRNA will be translated into proteins to carry out functions. So whenever we are doing translation, we need tRNA, which serves as an adapter molecule between an affinity for, because it has an affinity for a specific amino acid. In initiation, we have in prokaryotes a Shine-Delgado sequence, which is present for the attachment of the small ribosomal subunit. In eukaryotes, the initiation complex will move along the mRNA until it finds an initiation codon, AUG. AUG is helped identified by the Kozak sequence. During elongation, we make the protein. A charged tRNA will enter the A site, and a peptide bond will be created between the amino acid and the A site and key sites. The ribosome will translocate to the next codon, which will be 3 nucleotides down. During termination, the process is ended, and that's when a stop codon is reached, because there's no anticodon for a stop codon, so the polypeptide chain will be released.
I just have one more question for you. What do you think is the most important for the continuity and prosperity of life?
Well, we must go back to the fundamentals. The central dogma of biology says that DNA forms RNA, which forms proteins. Transcription and translation occur between those steps. And of course, for the prosperity of life,